The eggs are actually, when they're retrieved, they're actually not round like what we think of when we think of human eggs. They're actually surrounded by cumulus cells, which I think we talked about last time. So it's almost like a small dot, which is the egg, surrounded by a big cloud of white cumulus
Researches
cells.
are being done, papers are being done on these topics.
Yes, there have been research projects about this for a long time, and really there's just no definitive answer because there are just a lot of factors, like we just talked about. There are so many factors that go into play. So it's really hard to say for sure if it's one specific thing that's causing it.
Yeah. It's fascinating because we see so much development and so much progress, and then you come back to a small thing and there's so many divided opinion by experts and there's no concrete proof. Wow.
Yes, it's a little frustrating in the field because wherever you look, there are conflicting results and it's hard to know what's true and what's not, or are they both true?
Yeah.
Or, it's hard to have definitive answers.
Yeah. They could both be true under different circumstances.
Exactly. Yes. And what one clinic sees might be different than what another clinic sees. And there are just so many variables that are at play all the time. So there are just so many variables that go into every calculation and every result.
All right. Well then, thank you so much, Jessica, for your time. And
Absolutely.
I look forward to seeing the email and editing team will work on that.
Yes, I will send that email over to you now with those videos. And if you don't get them, let me know. And then, we can talk. Maybe next time we can talk a little bit about embryo grading and embryo freezing and thawing. And maybe then another time we can talk about PGT because that's a pretty complex topic.
Yes. And I think in many countries it's against the law.
Yes, I don't know all of the laws specifically, but I do know that it's not permitted in some countries. Knowing the gender is not permitted in some countries. Gender selection is not permitted in many countries. It's a little bit of a controversial topic, but one that's still performed nonetheless. So.
Yes. Well, thank you. You have a very nice evening, and I will see you Monday evening at 8 p.m. your time.
Okay, that sounds wonderful. Thank you.
Bye.
Bye. So, like today, I plan to talk about an egg retrieval and insemination. And I have videos for both of them. And I think that those videos are actually probably better than a presentation because they actually show you the processes themselves.
Exactly.
So the embryologist job really starts at the point where eggs are retrieved from a woman's ovaries. So to back up just a week or two, a woman's IVF cycle will begin at the start of her menstrual cycle. So at that point, she'll come in for baseline ultrasound monitoring to make sure that she's ready to start her IVF cycle. From there, she'll begin to administer medications that help the eggs inside of the ovaries mature. Normally in a menstrual cycle, multiple eggs begin to mature, but only one ultimately matures while the others degenerate. But what these medications strive to do is have multiple eggs mature instead of just one, so that multiple mature eggs can be retrieved instead of just one.
Giving the client a better chance of getting pregnant.
Yes, more eggs equals more chances of fertilization, more chances of embryo development, more chances of a pregnancy.
Correct.
So, right, more mature eggs is better. So there are a wide variety of how many eggs women can have retrieved. For example, women with PCOS might have a larger number of eggs retrieved, whereas women with diminished ovarian reserve might have a lower number retrieved. The number is important in a sense, but really what's important is how many good quality embryos develop from the eggs that are retrieved. So that's one of the primary focuses of the medications. The other is to control the timing of ovulation. So we want the egg retrieval procedure to occur right before ovulation occurs. If it occurs after ovulation, the eggs will actually be expelled from their follicles in the ovaries, and then they can't be retrieved. So we want the egg retrieval procedure to occur right before ovulation, but we want to give the eggs as much time as possible to mature before they're retrieved.
Right.
And so every couple of days, you'll come into your IVF clinic while you're administering your medications. They're called stimulation medications. And your doctor will perform an ultrasound, and they will measure the size of the follicles and the ovaries that are growing. So eggs sit inside of follicles in the ovaries. Follicles are fluid-filled sacs, and they'll grow as the eggs inside of them mature. So they'll monitor to make sure that those follicles are growing appropriately. And then once those follicles reach a certain size, your doctor will administer a trigger medication, and this triggers ovulation to occur. But what happens is the egg retrieval procedure is scheduled for about 35 to 36 hours after the trigger medication is administered. And again, this is to get the eggs before they're ovulated, but once they're very mature. So at that point, you'll have your egg retrieval procedure where the doctor will go into each follicle in the ovaries that is grown and will aspirate or suck up the fluid. The fluid is then taken to the IVF lab, and hopefully it contains an egg. So hopefully each of the follicles that developed will contain an egg. Sometimes they don't, but if they do, the embryologist will find the eggs and they'll put them into a dish, a separate dish. So in that separate dish will be all of the eggs that are found at the retrieval, and then all of the other fluid can be discarded because at that point it's not needed. So we'll take those eggs. The eggs are actually, when they're retrieved, they're actually not round like what we think of when we think of human eggs. They're actually surrounded by cumulus cells, which I think we talked about last time. So it's almost like a small dot, which is the egg, surrounded by a big cloud of white cumulus cells. And so what we do to make the eggs easier to transport is we'll actually cut some of those cells from around the egg. So we won't cut the egg itself, just some of those extra cells, and that helps us keep track of the eggs as we move them. So we'll do that. And then some clinics will actually go ahead and will clean the eggs completely right after the egg retrieval. So they'll put the eggs into an enzyme that actually helps to remove those cumulus cells from around the egg. And then what you end up with are those nice bald eggs, we call them. So they are completely round. They don't have any cells around them. And at that point, the eggs can be graded based on their maturity. So before I get into
where grading comes in.
A little bit. This is a little different than the embryo grading. This is what we call egg grading. And before I get into that, I will just say that, and we'll talk a little bit about insemination in a few minutes here, but there is one type of insemination called conventional insemination or conventional IVF. And in that case, the eggs actually aren't cleaned all the way. So there are situations where they won't be, but if ICSI is being performed or the eggs are being frozen, those eggs will be completely cleaned. So there won't be any cells around them. So then once those eggs are clean, we can actually grade them based on their maturity. Eggs are one of the only cells aside from sperm cells that divide via meiosis, and it's just a type of cell division that eggs and sperm go through. And what happens is it ends up with a cell with only one set of chromosomes. So I won't go too much into detail about that, but I will say that meiosis is a long process. It actually starts before a female is even born, and it halts. It stays completely stopped in development until about puberty. And then once puberty occurs, you'll notice some advancement in meiosis for some of the eggs. Not for all of them, but just for some. But then the process will halt again. So in order for an egg to be inseminated and to fertilize properly, it actually has to complete meiosis 1. So there are 2 cell divisions in meiosis, meiosis 1 and meiosis 2. And cells cannot be fertilized unless they have completed meiosis 1. So this is where egg grading comes into play. We want to make sure that eggs are mature before they're inseminated or before they're frozen. And we know that eggs are mature if they contain a polar body, which is just a small little cell that almost sits inside of the shell with that egg. And if we see a polar body, we know that the egg has completed its first meiotic division or meiosis 1, and that it's able to be inseminated and achieve fertilization. So once we know how many mature eggs we have, and something to keep in mind is that not every egg that is retrieved is mature in many cases. So we expect on average about 70% of the eggs that are retrieved to be mature. And there are a lot of reasons why eggs may not mature. For example, some may not respond properly to the medications, or in some cases they just don't develop properly for any number of reasons. But regardless, we do expect about 70% 70% to be mature. And those are the eggs that can be frozen or can undergo ICSI. Again, for conventional or standard insemination or IVF, we do not grade the eggs because we don't clean them the whole way. And that's because those cells that surround the egg whenever they're retrieved are actually needed for that, for conventional or standard insemination. So either way, a few hours after the retrieval occurs, the eggs can be inseminated. Again, there are two types of insemination. There's standard or conventional insemination. It's also called IVF. And in this case, the eggs that are not cleaned are placed into a drop of media with a washed sperm sample. And they're placed together overnight in hopes that the sperm will be able to fertilize the eggs inside of those drops. The other type of insemination is called ICSI. It's ICSI, and it stands for intracytoplasmic sperm injection. And with ICSI, individual sperm are actually chosen and are individually injected into each mature egg. So one sperm is injected into one mature egg. And ICSI is indicated whenever there's any type of male factor infertility. So if there's a low sperm count or a low motility, or maybe there's a lot of sperm that are abnormal in appearance, ICSI is preferred. And that's because the embryologist can actually choose the sperm that are inseminating the eggs. Whereas with the other type of insemination, we have no control over which sperm will ultimately fertilize the egg.
Yeah, so it's a more deliberate method.
Yes, it's almost like a forced fertilization in a sense. We force fertilization to occur.
And does this give better result?
Yes, overall, ICSI does tend to yield better results compared to conventional insemination. But one thing to keep in mind is that eggs are graded prior to ICSI. So only mature eggs undergo ICSI. Whereas with conventional or standard insemination, the eggs aren't graded beforehand. So it's possible that some of the eggs that are placed into the drops with the sperm are not mature, so they won't fertilize. So we do expect overall better rates with ICSI, but that's the primary reason why.
So we're talking about an entire world, but this is just 1 or 2 drops of culture, right?
Yes. A couple of drops, really. Normally we try to keep 5 or less eggs per drop of media. Yeah, so it is.
It's pretty fascinating.
Yeah, it is. And it takes a little bit of time for us to be able to tell if fertilization actually occurred. We can't tell right away. We actually let the eggs that have been inseminated sit overnight in the incubator, and we'll check them the following day. Usually it's about 16 to 19 or 20 hours after insemination occurs that we check for signs of fertilization. So normally it's the following morning after the egg retrieval and after insemination has occurred. And what we're hoping to see are 2 circles inside of those embryos. Those are called pronuclei. And they're basically the nuclei of the sperm and the egg that are coming together to form the nucleus of the embryo. And the nuclei contain the genetic material for the sperm and the egg, which will come together to create the genetic material for that embryo. So an embryo will actually contain 2 sets of DNA, one from the sperm and one from the egg. We also want to make sure that there are 2 polar bodies present. So after the retrieval, we were looking for 1 polar body, which indicated that the egg completed its first meiotic division. After insemination and fertilization occur, the egg actually completes its second meiotic division. So we expect to see 2 polar bodies. So 2 pronuclei and 2 polar bodies means that the egg has been properly fertilized.
Wow.